Okay, perfect. Let me try that. Can people see my slide?
Close. It's not full screen?
Yeah. How's that? Is that working?
No. Okay, give us a moment.
Okay. I have it on full screen on my screen, so.
Okay, why don't we put up your slides on this end and you tell us when to advance?
Okay, sure. That's fine. Okay, so let me know when you have that up.
We are ready.
Great. Okay, so you know, I apologize first of all for not being able to make it in person, but I think this is really a fascinating workshop, and I really appreciate the organizers for giving me the invitation to speak about something that we kind of really recently described in a paper in Science, and it's about this utilizing the CRISPR-Cas technologies for recording biological signals directly into CRISPR arrays. And so I'm at the Department of Systems Biology at Columbia, and let's get started. So slide 2, I guess. One key area in this is really that biological signals are really difficult and challenging to record in complex environments. For example, in the gastrointestinal tract, in some in vivo setting where it may be very difficult to have a probe that goes directly into that environment, as well as the variety of different metabolites and toxins that one might be interested, especially in the context of this discussion. Next. And also in addition, there's tracking of dynamic regulatory programs, such as things through development that one might be interested in kind of sensing over time, over a longer time scale. And so if you click through to the next figure, that it basically the current limitation is that you know, these imaging and recording modalities really require us to do continual data acquisition, right? That these signals are basically present on a very short time scale, and you have to capture that signal right away for you to be able to understand and know what that signal is. And if you don't capture it, then you lose that signal over time. And so what we really need is something that's untethered. It's an autonomous surveillance system that you can interface with the natural cellular system in its native environment as it's performing some recording, almost like a submarine as it goes down into the ocean, and it can kind of surveil kind of the landscape without having to be tethered to anything else. So the next slide just describes the kind of the recording, you know, what has been done historically, you know, in terms of recording temporal signals, which is, I think, is the most challenging aspect of biological recording, is that how do you delineate when the signal occurred in time, and how do you distinguish that from other signals that might be occurring? And so people have, you know, done a variety of things, such as kind of physically etching on a medium, kind of the temporal signal as the kind of the medium is being linearly kind of rolled out. So in this case, an auto, you know, phonograph or a phonograph from Thomas Edison, you know, kind of there's a vinyl record player, right? So, kind of the next slide is really, you know, we really want to kind of convert this into something that is equivalent to, you know, what I would think is kind of this tape recorder. So, you know, people might be familiar with tape recording. So essentially the idea is that you can spool this physical medium through, and as the medium spool through the head, the signal is recorded digitally into that substrate, and you get directionality of the temporal signal by the result of linearly spooling the substrate here. So next slide. So really kind of, you know, what is the medium for which we could do this? You know, DNA is really a natural medium for doing kind kind of information storage in biological systems, certainly in natural biology, but you know, over the past several years, people have really begun to also take a keen interest in using DNA as a synthetic information storage medium. So if you kind of just click through all the animation slides here, really kind of DNA is, you know, in contrast to semiconductor kind of information, really has this kind of nice property that it lasts a really long time, potentially tens of or hundreds of thousands of years. It has very low kind of power usage, and information density is super high, and people have started to record actual digital information directly onto DNA by encoding bits and synthesizing the DNA from scratch. So therefore, kind of DNA information storage might be a really good medium for this type of biological information encoding that we're interested in. So the next component here is how do you build a memory system? And so the next slide really describes kind of you know, what we've been talking about over the last couple last day is you know, the CRISPR-Cas system. You know, most of the discussion has been focused on the CRISPR immunity component, which is the Cas9, kind of the nuclease function that is used to do genome editing. So what we're focused here is really on the acquisition component, which is the first component, which is the immunization component. That is when transient kind of DNA, such as viruses or plasmids, are introduced into the cell. The Cas system is able to capture those DNA elements and incorporate them onto the CRISPR arrays as spacers. So if you click through all the animations on the slide here, on the right side, you'll see essentially the current understanding of the mechanism of how this works. Essentially, you have kind of foreign DNA that comes in. The DNA is chopped up based on the position of the PAM sequences into protospacer sequences that then get incorporated into the array from a 5' to a 3' directionally right downstream of the leader sequence. And so therefore, this gives you a very specific position in which the new spacer sequence, which is a new element that's the cells has been exposed to in a very directional manner. So it's always putting the new sequences at the 5' region of the CRISPR array. And so next slide is really kind of describing the system that we kind of developed to utilize this natural CRISPR immunity immunization aspect we call trace, which is temporal recording and arrays by CRISPR expansion. And so the idea is that if we can link the induction of a copy number inducible plasmid with a biological signal, that signal could then be preferentially incorporated into the CRISPR arrays compared to the background sequences. And because the background sequences, such as things coming from other plasmids that are already present in the genome, in the cell, as well as the genomic DNA, gets incorporated at some very low level, you essentially have a difference in signal versus background, which constitutes kind of the temporal recording modality for this. And so kind of the key elements here is really that it's a unidirectional spacer acquisition, so we can get the time component. And because we have reference DNA sequences, we know essentially they constitute kind of time tick marks in time, so that we can delineate when something was triggered and when something was not triggered, and the duration of time in between triggering events. So not just the order of the triggers, but the duration in between trigger events. Next slide. So, you know, we first explored whether we could essentially associate the change in copy number of this plasmid, which we called the trigger plasmid, pTrig, so that in its uninduced state, it essentially is replicating a very low copy. And under a induced state by a environmental signal or a change in gene expression associated with the sensor of interest, it increases the copy number of this pTrig. And so this is essentially by inducing the plasmid, it does a self-auto kind of amplification of the copy number in a very short period of time. And so you can essentially show, based on kind of quantitative PCR, that as you increase the level of the induction, you can get several orders of magnitude amplification of the copy number of this replicating plasmid. And so on the bottom of the slide here is just showing a titration curve of the induction of IPTG, which in this case is what the pTrig plasmid is sensing, and the copy number associated with the pTrig plasmid. And so this shows basically that you could link any biological signal to amplification of a piece of DNA. And then the next step is really, can we then capture that piece of DNA by the natural Cas1-2 system? And so kind of next slide, and we can kind of, why don't we just click through all the animations. Basically, we start out with a population of cells, we induce with an inducer, and then we recover the population. And as the cells are recovering, there's an there's this kind of transient recording of the induced plasmid. And so so basically the p-triggered plasmid is activated by the presence of the signal. That leads to an increase in copy number, which gets recorded by another plasmid, which is called the pRec, which is the recording plasmid that it expresses the Cas1-2 proteins, which are the only necessary proteins in E. coli that is necessary to incorporate spacers into the expanding CRISPR array. And so on the bottom right side, you'll see basically the an illustration of what a natural array looks like. As you're incorporating new spacers, you can essentially get three types of spacers: either genomic DNA, the recording plasmid, which is essentially also genomic DNA in essence. So those two both correspond to reference sequences, and then the trigger sequence, which is outlined in orange here. And so the bar graph in the center there just shows when you have both the recording plasmid on as well as the triggered sequence with the induction of the IPTG, you get a drastic increase by essentially several orders of magnitude of the spatial incorporation of the pTrig plasmid, which constitutes a signal. So we kind of can demonstrate that the array can be synthetically induced to capture new sequences. Slide 10 is the next slide really is to try to, then we start to explore basically, we can maybe click all the way through here, whether we could start to record biological signals in the time domain. So we essentially, instead of just doing an induction or not an induction, we do it over a series of days, in this case, over 4 days consecutively of the same population of cells. And so in this illustration, we basically have no signal, activation, and activation. So it's off, on, right? And then we can and then look at what the array expansion looks like by just directly sequencing the array. So I won't have time to describe, but essentially we come up with a method to use Illumina next generation sequencing to sequence the CRISPR arrays of the population kind of very efficiently. And so the first thing you can see is basically that the magnitude of the recording corresponds to, we could kind of essentially get magnitude of recording, which is the number of times the signal is present is proportional to the percentage of the pTrig spacer that's acquired. So it's basically a counter of the number of times the signal can be found, right, regardless of temporal information. And then the second component, which is the temporal recording, is do we see the position of the incorporated arrays to be present in, at the right spatial location to where they were actually induced. And so if you just look at all the arrays, on the bottom there's an illustration of 50 different arrays, and you can see that the induction profile matches that of the kind of resulting array kind of profile there. And so again, what I'm displaying actually is inverted in that you're starting from position 4 is the most recent array, position 3 is the next one, 2, 1. So the most distal position in the array is actually the earliest time point of the recording, and the most proximal one on the 5' end is the most recent. So the next slide is we then basically did all combinatorial mapping of all different configurations. There's 16 possible ways in which we can induce this array over the course of 4 days. And so just by kind of looking at this, we can look at first when everything is off, all the arrays are essentially off. And so what I'm showing here is basically the average percentage of pTrig spacers across the population for different lengths of the array. So within the population, you have both arrays of length 1, 2, 3, and 4, 5, and so you can look at any of those lengths and kind of extract information from that. And so basically what you see is that when it's off, everything is off. When one of them is on, at the most earliest, at the latest time point, the most proximal array position is triggered, and when the signal is kind of triggered at the very earliest time point, the most distal array position is basically being recorded here. And so if you want to kind of click all the way through here, you could basically see that they actually visually map to what you would expect, right? So you could get essentially a profile of what the signal looks like basically by just looking at the arrays and the frequency in which these spacers are being incorporated. So on the right side is just an illustration showing what the profiles look like for a length 4 array, and then what the actual frequency, the distribution that we actually find for each of the populations. And so you can kind of visually already appreciate that the information is captured there. So next slide is really, so then we kind of ask, could we kind of get more quantitative about this? You can actually map what the possible configurations, array types that could be generated through this type of differential induction profiles. And so we built a very simple kind of 4-parameter model of essentially CRISPR array expansion. And this allows us to basically model what the different types of profiles look like. So if you look through the animation, whether it's off, off, on, or on, off, off, what the expected profiles should look like across the entire population. And so, and I won't go into too much detail, is to say that the actual kind of model looks quite similar in terms of predictions as what the data looks like. And really to fit this with the model, you only need to kind of four very simple parameters. So next slide is really, then we kind of evaluated, let's say that we just evaluated the arrays by themselves. Based on the models, can we predict whether we can extract the temporal information directly from the array sequences, and how do they match the actual, the data that was generated and the actual temporal induction profiles? We can see that basically there's a perfect match if you integrate all length 2 through length 4 arrays. If you only use length 1 arrays, which has no temporal information, you get basically a quarter of the time being correct, right? Basically, it's a magnitude sensor. If you incorporate, information about just a length 2, length 3, or length 4 arrays, you do reasonably well, but not perfect. But if you incorporate all of those arrays together informationally, you get kind of the best array prediction. So next slide, just, we also show that this thing is actually quite stable, so you can record this over long periods of time. So we basically took a recording population, induced it over, and then just passed it over a series of 8 days, and the array information is kind of minimally disrupted over time. So finally, we then asked, could we then actually multiplex this, right? So could we do simultaneous recording of multiple different signals at the same time? And in this case, we took 3 different sensors. So again, these basically, the system is actually quite modular. So you could take any biosensor that has a transcriptional activation domain, such as a promoter, and put that upstream of the replicative gene associated with the pTrig sensor, and then that will basically become a sensing unit for the system. And so we have one for copper, for cellulose, and as well as for fucose. And so, and then if you click through the animation, we ask, could we also do simultaneous recording of all of this? So we basically have all 3 different signals recorded across a population. There are 3 individual types of recorders within the population, one that senses copper, one senses cellulose, one senses fucose, and we can uniquely distinguish between all 3 by doing a very simple mutagenesis of the inverted repeat, the direct repeat sequences in between the spacers. And so those are naturally conserved. So if you make mutations on the 3' end, you can essentially not change the function of the direct repeat, but generate unique barcodes. And so basically we can barcode different populations of sensing systems and pool them all together to do simultaneous recording. And so the next slide is just showing that by doing, and maybe we can kind of click all the way through, by doing different induction profiles of different combinations of the 3 chemicals, you could get activation of all 3 different sensors in the correct kind of combinations, right? And again, this is in a mixed community where you can do this. And so in the next slide, we kind of then ask, okay, can we start to actually then do what we were doing before with doing temporal recording of the system? And so basically you have on day 1 in this illustration here, fucose, cellulose, and copper all present in the medium. And then on the second day, only fucose and cellulose is present. And then on the third day, only fucose is present. And so you expect that the sensors for fucose, which is GAL-S, is basically activated all the time in the system, versus the sensors for cellulose only activated in the first 2 days, and then copper is only activated on the first day. And then you can then do some classification based on the model that we've described for this. And so on the next slide here, is just a summary of the accuracy of doing multiplex signal recording. And so we actually do reasonably well. That, of the 16 different profiles we tested out of 512 possible different profiles, we can get kind of very accurate kind of recording of most of the biological signal. So kind of I just want to wrap up here to say that I think there's a lot of really exciting opportunities for using this type of system, not just for recording, but also potentially for other things. So for example, you could potentially combine this with some type of actuation system, such as a ribo switch, or kind of one of these toll switches, that you could then potentially do a computing of the spacers based on the temporal orientation, temporal kind of ordering. You could potentially interface with the other kind of mediums, such as kind of digital signals, as well as a recording a variety of different environments, such as in the gut, as well as kind of recording a variety of different endogenous temporal transcriptomic signals. So with that, I'd like to just thank people in my lab, as well as kind of the major people who have kind of did this work. Robbie Schetz, who's a really talented graduate student, as well as Sung, who's a postdoc in the lab, and Felix, who was a rotation student on the project. With that, I'm happy to take any questions. Apologize for the kind of audio-visual technical difficulties.